We're still in chapter 26, and this is going to be on electrolyte balance, focusing specifically on sodium. But first, we will talk about what electrolyte balance is. And usually, this is going to be referring to salt balance, even though, as we went over a couple of videos ago, electrolytes also include acids, bases, and some proteins. Now, salts do a number of things. They control fluid movements, provide minerals for excitability, secretory activity, and membrane permeability. And they enter the body by ingestion and metabolism, and they're lost via perspiration, feces, urine, and vomit. These next two slides are a table or tables of different causes and consequences of electrolyte imbalances. Unless we specifically go over it, though, you do not need to know it for the exam. I guess it was one table. As you may remember from the first video, sodium is the most abundant cation in extracellular fluid. Sodium salts contribute 280 milliosmoles of the total 300 milliosmol extracellular fluid solute concentration. That is a significant portion, 280 of 300. It is the only cation that exerts significant osmotic pressure. It controls extracellular fluid and water distribution, and this is because of the fact that water follows salt. And changes in sodium levels affect plasma volume, blood pressure, and extracellular fluid and interstitial fluid volumes. When sodium leaks into the cell, it's pumped out against its electrochemical gradient. Sodium also moves back and forth between extracellular fluid and body secretions like digestive secretions. And then renal acid-base control mechanisms are coupled to sodium ion transport. There's a difference between sodium concentration and sodium content. So concentration of sodium is going to determine the osmolality of the extracellular fluid, and it's going to influence the excitability of neurons and muscles. It's going to remain stable because water shifts into or out of intracellular fluid. So the concentration of sodium remains relatively stable. The content of sodium is a total body content, which determines extracellular fluid volume, therefore blood pressure. So this is looking at sodium concentration and sodium content and the homeostatic importance. So the homeostatic importance of sodium concentration, again, is extracellular fluid osmolality, and the homeostatic importance of body content, body sodium content, is blood volume and blood pressure. Now, the interesting thing is that there are no known receptors that monitor sodium levels in body fluids. Even though it contributes 280 of the total 300 milliosmoles, there are no known receptors. Sodium-water balance is linked to blood pressure and blood volume control mechanisms, and changes in blood pressure or volume will trigger neural and hormonal controls to regulate sodium content. So we start off by talking about the influence of aldosterone and angiotensin II. So aldosterone plays the biggest role in the regulation of sodium by kidneys. Regardless of aldosterone presence, 65% of sodium is going to be reabsorbed in the proximal convoluted tubules, and 25% is reclaimed in the nephron loops. Sodium is never secreted into the filtrate. When aldosterone concentrations are high, sodium is actively reabsorbed in the distal convoluted tubules and collecting ducts, and water will follow. So extracellular fluid volume will increase. And that's because at this point what's happening is that we're reabsorbing the stuff. We're taking this stuff out of the filtrate, putting it back into the extracellular fluid. So extracellular fluid volume will increase. When aldosterone concentrations are low, sodium is not actively reabsorbed and it's lost to urine along with the loss of water. Remember, water follows sodium. The renin-angiotensin-aldosterone mechanism is the main trigger for aldosterone release. And then we go back to the granular cells of the JGC that secrete renin in response to these three things: sympathetic nervous system stimulation, a decreased filtrate sodium chloride concentration, and a decreased stretch of granular cells due to a decrease in blood pressure. Renin catalyzes the production of angiotensin II, which is going to prompt aldosterone release from the adrenal cortex. This is going to result in an increase in sodium reabsorption by the kidney tubules, again, in the specifically distal convoluted tubules and collecting ducts. Aldosterone release is also triggered by elevated potassium levels in the extracellular fluid, and it's going to bring about its effects slowly, so hours to days. So if we look at this, we have a decrease in body sodium content triggers renin release, increasing angiotensin II, which is going to stimulate the adrenal cortex to release aldosterone. This is going to target kidney tubules. It's going to increase, well, let's go this way. This way, we're going to increase sodium reabsorption, and then we're going to return back to homeostatic plasma levels of sodium. We go this way, we have an increased concentration in the extracellular fluid of potassium. Also stimulate the adrenal cortex to release aldosterone to target the kidney tubules. And this way we go this side, and this is going to increase potassium secretion, which is going to restore homeostatic plasma levels of potassium. Atrial natriuretic peptide is released by atrial cells in response to stretch, which is caused by an increase in blood pressure. And what atrial natriuretic peptide is doing is, well, its effects are to decrease blood pressure and blood volume. And it does this by inhibiting ADH, renin, and aldosterone production, increasing excretion of sodium and water, and promoting vasodilation directly, and also by decreasing production of angiotensin II. Female sex hormones also play a role in the regulation of sodium balance. Estrogens lead to sodium chloride reabsorption, and this can lead to water retention during the menstrual cycle and pregnancy. Progesterone decreases sodium reabsorption, and it promotes sodium and water loss. Glucocorticoids increase sodium reabsorption and promote edema. We have cardiovascular baroreceptors, and they will alert the brain to increases in blood volume and pressure. The sympathetic nervous system impulses to the kidney will decline under these circumstances. This will cause the afferent arterioles to dilate. It will increase GFR, glomerular filtration rate, and this is going to lead to an increase in sodium and water output, and that will lead to a reduced blood volume and pressure. So let's look at this. So we have a decrease in systemic blood pressure or blood volume, and under these conditions, there would be a decrease in filtrate sodium chloride concentration. This is going to cause the granular cells of the kidneys to release renin. Renin is going to catalyze the conversion of angiotensinogen to angiotensin I. That gets converted to angiotensin II. This does a couple of things. It's going to stimulate the adrenal cortex to secrete aldosterone, which is going to target the kidney tubules, which is going to cause an increase in sodium and water reabsorption, which is going to increase blood volume and blood pressure. We also may have it go this direction, and with this we have the angiotensin II causing the systemic arterioles to vasoconstrict, which will increase peripheral resistance, which will lead to an increase in blood pressure. And this can also occur because of decreased stretch in the afferent arterioles. Here are the questions for this section. And this is all I have for this video.